Hello, everyone. This is Senior Biotech Analyst John Vandermosten. Welcome to our channel that educates the life sciences investor on exciting advancements in drugs, biologics, and devices. For more content and news like this, subscribe to our channel below and like or share with others. So we'll continue on with our discussion of the RECOVER trial. And, I believe that you recently reported some new findings from RECOVER. You initially reported top line last fall, but just recently there was some other data that came out. Why don't you share some of that and get investors up to date with what the latest is on the data?
Yeah, in the top line data announcement, we disclosed a primary endpoint and then key secondary efficacy endpoints. Last week we had a KOL webinar in that we announced additional data, especially biomarkers data for a BDNF is a neurotrophic factor, is closely associated with most of the neuropsychiatric indications. And then also neuroinflammation. We have key biomarkers, pro-inflammatory cytokines reduced. And then BDNF in schizophrenia patients as well as in psychiatric patients, BDNF level biomarker reported to deplete. If the treatment works and the BDNF level should go, we have seen increase in BDNF level from the baseline to end of treatment, and then compared to placebo. That's a good sign. That means treatment effect is...
Because I think, isn't that somehow associated with neuroinflammation as well?
Yes. What we know so far in the literature, the patients who are chronic schizophrenia patients, they have depleted level of BDNF, and then that also increased level of neuroinflammatory markers. We call them pro-inflammatory cytokines. So if the treatment works, especially the drug has a anti-inflammatory effect, the BDNF level should go up than the baseline or the beginning of the treatment, and then pro-inflammatory markers a level should go down if the treatment works. That's what we have seen in our drug, significant decrease in pro-inflammatory markers, significant increase in BDNF. That's what we expect to see in an ideal situation.
So, and I think we got another biomarker too, prolactin, which was also important. It's a hormone that can have some unwanted side effects. Maybe you can explain what that is, and then also what we observed in brilaroxazine arm versus the placebo arm for that.
Yeah.
That
so
hormone.
The hormone called prolactin is associated with this hormone as well as another hormone, thyroid hormone. So these two are, known to have a, imbalanced in this schizophrenia patients as well as in general other neuropsychiatric patients, especially bipolar and then major depressive disorder patients. So schizophrenia patients as an organic cause of the disease itself, as well as some of the widely used antipsychotics currently in the market are known to increase prolactin level. So our drug decreased the prolactin. That is also a marker to some extent, the efficacy effect, treatment effect. So increase in prolactin is associated with number of problems in this patient population.
Like for example?
Increase in prolactin is also known to cause enlargement in breast, both men and women.
Okay.
And then also it's known to cause sexual side effects. Sexual side effects is a major problem in this patient population. So one thing to remember, most patients, the onset of disease in early teenage and then early adulthood, these are younger patients. So increase in prolactin in both men and women expected to cause sexual side effect. So this is a major problem in the treatment. Our drug not only reduce the prolactin level, that's the marker, and then also we have seen statistically significant decrease in sexual side effect, especially in females.
So much better side effect profile than other approved products.
Correct. Because in other words, you can consider prolactin decrease as a biomarker for improving or addressing the sexual side effect as well. So, and then also overall treatment outcome in decreasing the schizophrenia severity.